Hi, everybody. Welcome. My name is Dr. Rafael Gonzalez. I think we're going to wait probably about 30 seconds, a minute, um, just while everybody gets on, people start logging on. But anybody, welcome. Just tell you a little bit about my background for those that don't know me. I got my PhD from University of California, Irvine, where I worked actually in anatomy neurobiology at the Christopher Reeve Paralysis Foundation, specializing in spinal cord injury and the immune system. But I was part of the group that did, work back in about 2000 on stem cells and spinal cord injury. And then from there, I actually went into sector about 2006 and started studying various different stem cell types and other cells, actually other cell types, of which we are a US-based company that has been studying, cell therapies more than anything else, and spending a really, really big focus on reverting the immune system to a younger state, which is what our company focuses more than anything else. And then actually making yourself feel good inside, which is key to the immune system, maintaining immune health, and then also looking good outside, which hence is our product, JuveXO. So with that, I'll go ahead and start and so our company focus is basically just so a mission is, we consider ourselves a biocosmetic company because we've been, looking at cells and looking at things at the cellular level for many years. I mean, if you're going to see some of the data I'm going to show you, this data, predates 2015, 2014, where we started doing a lot of studies on this. But the cell source that we actually have, there's a publication back in 2010. So we're talking about 14 years ago. Besides that, you actually think of all the work that has to be done before that publication, you're now predating to about 2008. So it's a long time ago, quite a bit of work and quite a bit of development in all of it. And our vision is crystal clear on pioneering and setting the global standard in biocosmetics by providing diverse and continually innovative skincare products. For those that have heard me talk before, I always like to show this little basic slide because it's so important to understand, that cells are the smallest basic unit of life. Everything is built upon the cell in our body. If you think of the simple fact that there's over 300 billion cells that turn over on a daily basis in our body, it's pretty crazy. I mean, this has to happen, and it has to happen correctly on a daily basis. And one of the keys to this is direct communication from one cell to another cell, which are simply done through secretory vesicles and something called exosomes, which we're going to describe in a lot more detail. But the important part of all of this is understanding that the cells not only do release these exosomes, but they secrete out an extracellular matrix. And the extracellular matrix, which is consistent with collagen, and the most abundant one in our body, collagen 1 and 3, hyaluronic acid, elastin, fibronectin, that's secreted a lot by the 3D matrix that we have from fibroblasts also. These are key to health, and these are key to skin and looking at, changes as we age with skin. Now, the exosome, I'm going to show you diagrams so you completely understand what it is. These are different. Exactly what I just told you is that every single cell in the body communicates to another cell. It's not only stem cells, but every cell communicates to another cell in the body by an exosome. Exo meaning outside, and some is actually literal translation of body. And the way this is done is this is basically a nanoparticle because it's about 1/1000th the size of a cell, and it contains a bioactive molecule. But these are all different. For instance, if you think of a cancer cell, guess what a cancer cell is actually going to excrete out? It's going to excrete out a cancer bioactive message in the form of a nanoparticle that's going to go to another cell and possibly cause cancer, cause other things. These depend on the conditions. Even when we talk about when we put them in a petri dish, that I'll show you in a second, they can dictate what they secrete out, what they produce, in other words. These are all dependent on the age, the gender, disease, fasting state, cell type, medication, the culture condition, which is what we're going to show you a little bit of the work that we've actually done on this. And there's a clear definition of what the International Society for Extracellular Vesicles wants you to do to look at for you to demonstrate that you actually have an exosome or any form of extracellular vesicle. You want to look at either flow cytometry, nanoparticulate. You could look at electron microscopy or scanning electron microscopy, and there's other methods too. And then you want to look biochemically. Does it contain specifically at least 3 markers, of which you want to look at Alex, tumor suppressor gene 101. On the outside of this, you will find a protein called CD63, CD81, or CD93. Those are the predominant ones that we now know that are well characterized as a scientist that we can actually look at and we can say, okay, we firmly believe that we do have an exosome. So what is these exosomes? What do they carry? They carry so many different things. They carry proteins. They actually carry lipids. And they're actually made with a lipid bilayer on the outside. And the concept behind that is actually for one basic thing. It's a protective action so that the message can go from one cell to another cell without being destroyed. They contain messages like messenger RNA, microRNA, transfer RNAs, piRNAs, a bunch of different types of RNAs, even non-coding ones. Proteins, which proteins, of course, direct function. Please remember the message that I'm going to consistently preach to you guys is protein always directs function, not the message. It's a little bit different to get something in and cause it to make a change when it's a message. Now, these, as said, is a bioactive messenger, and you can see them clearly right here. You will see a cell, and first and foremost, it's an endosome because it's made within the cell. And then as it actually comes outside of the cell, as you see here, it's blebbed, and you'll see this lipid bilayer just like a cell, which protects everything that's inside of it. If you look at it under an EM, you'll see those images right there that are gray. Those are under electron microscopy. And now normally what you'll do is you'll disrupt that lipid membrane and you'll start reading protein, lipids, RNA, whatever you want to do. You want to look at it in the context of, are the proteins intact, which can be done by a Western blotting. You're going to look at the message. You can do that through PCR, polymerase chain reaction. And if you want to quantitate it, then you will run something that's called an ELISA. You can also quantitate them by doing a nanotrap assay, but those are flawed systems, and we'll talk about it a little bit more. Electron microscopy, where you can actually just visualize it, show it, and even a surfing plasma and resonance is another actually method of doing it. Many ways to test these things, but not that straightforward, not that easy to actually do, especially in the context of the way it's being done, what they call easily now, which actually just quantifying nanoparticles. And I'll show you some instances of it a little bit. Once again, an image, a clear image of understanding of this is released out of the cell, where you see the lower cell on the bottom there. And then it transports across. You can see it has different proteins on the outside of that lipid membrane. And then it goes to a target cell. This is not the case in every single message. Let's just be crystal clear on that. So cells also secrete out proteins, secrete out extracellular matrix. That's how we live in actually the 3D that we actually live in. It's because it's secreted out naturally. It's not only through an exosome or through direct communication from one cell to another. Cells also have tight junctions when one cell is right next to its partner cell, and it communicates directly through tight junctions when they're right next to each other. And in that instance, it also secretes out things to neighboring cells that can produce that 3D that we actually live in. This is a simple, fantastic diagram. One more time, just showing you in the context of an exosome being produced from a stem cell. You have this stem cell that's now trying to target and help an injured cell. And what you see is it can directly stimulate it, releasing an exosome, and directly bind to a receptor. You see the little red dot right there on the top left-hand corner that binds directly to it. And now it activates that cell to cause an action, to make a change. The other one on the bottom left-hand corner is now the exosome has receptors, and the receptors within the exosome, and it actually goes and binds and now transfers receptors to an injured cell that the injured cell can accept a message. Then on the bottom right-hand corner, direct functional change is it carries a protein that's protected. It binds to that cell, it incorporates the exosome into the cell and causes a functional change because it's a protein. And then the top right-hand corner one, it takes genetic information, messages, microRNAs, messenger RNA, carries it over to the cell, and it takes it into the cell. The cell has to incorporate it. And remember, most importantly, for a change to happen, it requires not only the message, but it requires an abundance of transfer RNA, and it requires the cell to accept it, that requires the cell to transcribe it into a functional protein. Otherwise, the change does not happen. So the protein is key to all this. Now, let's go into a little bit more. I just showed you a little bit about how they work. But I want to talk about the most important aspect of all this is where they come from. And what's the importance of where they come from? Once again, remember, every cell produces this in the body, produces an exosome. You can capture them easily. And in the context of stem cells, everybody's quite familiar with a mesenchymal stem cell. The mesenchymal stem cell, you can obtain it from bone marrow, fat, teeth, post-birth tissue, which is amniotic fluid, amnion, the placenta, the umbilical cord blood. You can capture them from there. All of these may help in growth and healing, but it depends on the condition, it depends on the donor, and it depends on the isolation. Simple example of such. If you guys decide to use amniotic fluid, people talk about I have an amniotic fluid product that they've captured. Guess what? From amniotic fluid, let's just say from a donor, they obtain 100 vials of it. Well, one baby is completely different than another baby that's completely different from another baby. So what is the signals that are in there that are consistent, which you don't know? Not only that, if you think of the context of an amniotic fluid, which is actually excreted out urine, something like amniotic fluid is mostly predominantly antibodies in nature. And what you see is you will register, a lot of times you'll see these with these machines, a registration of 1 trillion nanoparticles. It's because once again, it's reading nanoparticles. It's not registering the lipid bilayer. It's not registering an exosome. It's an incorrect read. I'll go into it a little bit more. Not only that, a lot of this stuff, what do you know about the genetics? What do you know about the viral panel? Yes, maybe they might test for herpes, they might test for mycoplasma to show you that it's clean, they might test for those. But what happens if there's a genetic inherent mutation within the exosome that was carried from mother, father? That's very, very possible. I'm going to talk about this a little bit again when we talk about cells. And what happens if the carrier has, a virus that you don't know about, a virus that's not tested, even though there's a specific panels of viruses that are not being tested. These are all things that you have to consider when you think of the production of these products, of products that contain, for instance, just umbilical cord ground up tissue. People are taking ground up tissue, just grinding it up, filtering it through, saying, here you go. You have exosomes in here. There's a bunch of particles in here. They're exosomes. It's got some growth factors in it. This is what you get. This is something that you have to be really, really cautious with. The other thing is that they're not all created equal, the mesenchymal stem cells. They're all considered multipotent. They can respond to specific signaling cues, but it depends on where the source is from and how potent they are. For instance, guess what? And I'm going to show you a little bit. If I took a bone marrow from a 40-year-old patient versus I took a mesenchymal stem cell from a bone marrow of a 40-year-old patient, I took a mesenchymal stem cell from umbilical cord, you have to think consciously that this is from a 40-year-old individual with inherent genetic issues, with inherent adult issues, with aging in general. And you can actually register this by looking at simple telomeres versus an umbilical cord that's something naive, fresh, that has a lot more potency, that has a lot more development and a lot more growth to do. You're going to be spot on if you're thinking this is a much better source. And not only that, depending on where it's from is what's going to dictate the signals and also the condition you put it on in. Simple example of research that we actually did. This is actually dated to 2014. If you look at this carefully, you have the diagram on the left of, just a simple example of you see some cells, round cells, and you'll see that pink, that's actually, it's media. That's not the type of media that we use because it has phenol red in it. And we don't use phenol red because we don't want to mimic estrogen. But you look at the cells that are stuck there, and the cells in there will grow and proliferate. And the cells, as they do so, they will release out exosomes, they'll release out matrix, they'll release out communication skills. Now, if you look at the bottom chart on this graph right here, you'll see U1, U2, U3, U4, and you'll see it says cell, and you'll see it says media. Media is the pink stuff that you see in the image right there, the picture. What I need you to understand is that each one of these cells, whether it was umbilical cord, our particular umbilical cord lining stem cells is what we call them, and I'll show you the publication in a second. It's either those or it's a bone marrow mesenchymal stem cell that's 40 years of biological age that we compared it to. And when you look at it in this context, we looked at capturing hyaluronic acid and chondroitin sulfate, both the two different things that work actually phenomenally well for skin, first and foremost. And you'll look at what's inside the cell. If you look at right here, essentially transitions into what's outside of the cell when you look at the media portion. And then we look at the context of what gives us the most abundance of what we're trying to capture. In this case, we picked this formula right here because it's the ideal one for the hyaluronic acid, and it gives us a good balance of chondroitin sulfate. That's the formula we picked. We put it in various different formulations, and we decide. We actually dig a little bit deeper in understanding of what we wanted. But let's get clear with what you're hearing out there and what everybody talks about: exosome, exosome, exosome. The vast majority of everything that's out there is either a conditioned media or just a raw secretome product. And a raw secretome product is something what I just told you right now. Somebody just takes amniotic fluid. They basically just filter it through and they give it back into you. I want you to, the reason why I put this diagram out there is so you clearly understand. It's very, very, very difficult to isolate a pure exosome. And it requires this very, very last step that's on bottom here. So when you start off with culturing cells, if you go with that route on culturing cells, which is the best way to purify a stem cell, because once again, if I take bone marrow from somebody, the amount of stem cells that are in there are 0.0000001% in an average 40-year-old person. If I do it from fat, 0.00001%, it's not that many more that are there. But when I culture them, I purify the stem cell, and I now am giving you a purification, a pure stem cell. So I produce a pure mesenchymal stem cell. In the case of ours, we call it an umbilical cord lining stem cell. And then from there, I put them in a condition, which you'll see right here, the very, very first graph, the stuff that's on top of those Petri dishes, those dishes there, those are called flasks. And then from there, if I take just a very, very top portion of it and just collect that while I was growing it in, that's a conditioned media. That means that the cells were conditioned, the media, excuse me, the food that we feed them was conditioned by these cells. Then all I did was isolate that, and I can give you that. You could say that's an exosome because there's exosomes in there. But in theory, there's a bunch of other stuff in there, and it's expelled, recycled food from the cells. If I go a step further and I start purifying it by centrifuging it down, then I then get a secretome, which basically gives me a little bit of everything. There's exosomes, a lot more purified exosomes in there, but there's also a bunch of different factors in there depending on the cell type, depending on the way you grew it up. And then it captures that portion. That's what we call a secretome. And depending on the way you make that secretome, you can purify it even further, which are some steps that we do with an ultra purification step. But then if you really want to capture just pure exosomes, you then target the markers that we talked about earlier. The best known separation techniques are using CD9, CD63, CD81. These are proteins on the outside of the cell. I can attach a bead to it. The magnetic bead then pulls only those, or it negatively selects for other population types. And then I capture that. When you do that, you'll be blown away how much lower, how many less total exosomes you actually truly have. So what's out there? We talked about amniotic fluid are in there. And amniotic fluid, if you think about it, it's really not abundant in stem cells. It's a filter, once again. It's a barrier and it's a filter, and it's mostly epithelial cells. Remember, the filter in our body the cell type is an epithelial cell. There's umbilical cord ground up tissue. Many people have them. They talk about their secretome, their exosomes. Very inconsistent because remember, donor to donor variance. You have one donor, you're going to produce 100, 200 vials from one donor. Then you have to go to another donor, then another donor. Every single one of us that are on this call are completely different. Our kids are completely different. Which is neat about biology. Bone marrow mesenchymal stem cells. Only problem is the older donors. Let's say you get it from somebody that's 40, and then you actually grow them up and you age them. Show you the data on this, and a lot of them lack a lot of testing. Placenta is a filter, once again similar to the amniotic and to the amnion. Not abundant in MSCs. You have to isolate the MSCs from there, and it's consistent with being a filter. And once again, it's something that you cannot grow up in abundance to produce this master bank, master working bank. It's not an easy task in there, and the amount of testing that's needed. Plant-based technology, I hate when people come up to me and say, you're transferring human tissue from one to another, transferring human genes. You can cause infection, you can cause disease. Well, guess what? You can get sick as sick can be with plants. You could have transfer of yeast and fungal, which is now transpiring like crazy, the amount of infections that are out there now. You can get bacterial infections from plants. And once again, the most important aspect of it, how effective is a plant that contains a plant cell wall, not a cell membrane, to that of a human being? They're different protein sequences. So how efficient is that going to be? I do not use, when I grow up cells, human cells, I don't use plant growth factors or plant cytokines. We use human recombinant ones, is the way to go. Fat cells are pro-inflammatory in nature. They work in some instances. But once again, you have to obtain fat, you have to grow up the cells, and they become old. They do become old. It becomes an issue. Our product is derived from the umbilical cord lining stem cell. This publication is dated from 2010. And if you think of the umbilical cord, the lining of it, it's the conduit of life. It basically plays a crucial role in sustaining life. It continues to sustain youth and any form of natural healing. And the neat thing about it is, think of this. It's the one thing that has to have baby, mom, and dad. How is baby not rejected when it has dad's genes in it and mom and baby are connected? It's because of the immune privileges that this has, that this carries. And it's able to transfer nutrients back and forth from mom to baby, and actually vice versa, antibodies, all of it, vice versa, which is pretty neat how it does so and how it sustains life. We look at comparisons because we're all about making sure that we're giving you the best product that's out there. And one of the keys when we look at cells is we look at, number one, something that's called a colony forming unit assay. It's essentially a measure of the ability to clone or produce from one cell another cell, which basically now defines a stem cell, stemness. If you look at it on the top panel, the bone marrow, you'll see in 4 of those plates, what we do is we only put in one of those plates 100 cells. And then on the plate that looks darker, those are 10,000 cells. When there's more cells, you're going to be able to, there's more cell-cell communication. Cells are happier. They're going to grow. They don't have to clone as much. They can grow easier. But what you'll see is we then quantify the dots. The black dots after 14 days is what we quantify. And that gives us this colony forming unit assay where we can say, this is the frequency of sort of the ability for one cell to clone itself. In the fat, you look at the same thing. And then on the very, very bottom panel, you'll see how many of those dots are there. That's the umbilical cord lining stem cell, where we now graph it out for you. And you can see the frequency of, as you age these cells, how much they can clone themselves. And even at the later time points, which you'll see the number 6, that's for the many times as we remove them from the plate and replate them, replate them, you'll still see that the stemness of the umbilical cord lining cells is far superior to even the initial start of the fat or the bone marrow. So saying, the same person's fat and bone marrow. The most important aspect of all this is looking at this in the context of measuring telomeres. The product that we produce, the product that we actually have in clinical studies right now, is what you see, this P4 product. And if you look at the context of what that is in 8,000 base pairs, if you look at biological aging, it's in the range of about 15 years of biological age. Because what transpires is you look at telomeres. So when egg and sperm is put together and an embryo is conceived, you basically start off with 15,000 base pairs. In the duration of those 9 months, 5,000 of those base pairs are actually cleaved. That's how much development transpires. That's how much cell division and aging transpires in those 9 months. 5,000 base pairs. If you look at the start of this median of telomere length, you'll see 10,000. You will start off when you're born with 10,000 base pairs. Throughout your lifespan, which you can test this, you will start cleaving telomeres. And the mechanism to protect that is an enzyme called telomerase. But you'll start cleaving telomeres, cleaving base pairs. At some point, if you start reaching this point, like you see right here on this P6 of this fat and this bone marrow, you're getting down to 6,000 base pairs. Guess what? You have critically short telomeres, especially when you hit about 5,000 base pairs. You are heading towards a neurodegenerative disease, a heart disease, some form of disease of aging that you better now make changes in your life to change this or find a way to reverse this. If you look at the blue bars, the umbilical cord, even at this later time point, that's when the cells have been duplicated out quite a bit. They're still much higher than that of a 40-year-old bone marrow mesenchymal stem cell or a fat stem cell from a 40-year-old. Safety, safety, safety is our key focus on everything. We've tested these cells extensively because, once again, in clinical studies, they're in clinical studies in heart disease, in heart failure, in autoimmune disease, and the COVID study in Duchenne's muscular dystrophy, and then going into many other studies relatively soon. Extensive viral and bacterial testing, and this is the most important aspect of it. I never knew until when we started doing testing of this. We have to test for parvo. We test for parvo, which is pretty interesting. I thought parvo was only transferred, within dogs, but essentially humans can get it. And the most important aspect of it is genetic testing. Doing the genetic testing, meaning we grow up cells and we test them at various time points as they age until they are aged completely. The concept behind that is to look for a chromosomal aberration, a genetic deficiency. When we went through 50 cohorts to find the 3 that we use that were produced prior to COVID, when we went through all of these, we only have 3 lines left over. We found many that had chromosomal aberrations. Where we would grow them up, we would find an aberration. We thought it was a perfect cell line because it was growing according to our dynamics, the quickness of growth, the quickness of when we obtained it, the donor screening, all the other testing, all the work that had been done. Then all of a sudden, boom, we get hit with a deficiency, chromosomal aberration. We then go back and test one more time to confirm, because guess what? We have to be the bearer of bad news, call the parents and say, we found a deficiency here as we grew these up. You need to go back and test mom. You need to go back and test baby. 100% of the time that we've done this, it's been spot on. They will go back and test baby. They'll go back and test mom. There's going to be a deficiency during development for the child. And the good thing about it, actually, it is bad news, but the good thing about it is at least the parents got a notification of what can you do to help with that change, what needs to be done to help with those children. Skincare is our focus. We want to look and feel younger with it, and that's actually JuveXO. It's designed for success. Number 1, contains collagen. People underestimate the extracellular matrix in your body. Collagen 1 and 3 are the most abundant proteins in our body. As we age, the ability to produce collagen decreases significantly. By the time you hit 60 years of age, it's decreased to over 50%. That's why we have wrinkles. That's why we have sagging skin. It's one of the reasons on it. And it's one of the proteins that actually strengthens many tissues in the body, cartilage, bone, skin, all of it. There's 16 different types of collagen. We're going to talk mostly about the predominant one. That's 80–90% of it. That's collagens type 1 and 3. And it's the most important for skin because containing about 19 different amino acids in it. Secondarily to that, and it's not a protein. Hyaluronic acid is not a protein. It's a proteoglycan. It's a GAG, and it's the second most abundant thing in our body. And if you think of where you would predominantly find it, is you find an abundance of it actually in our joints. It actually serves the cushioning in our joints, a liquid that's in a joint. And there's a big difference between it being high molecular weight and low molecular weight. As you would think, the high molecular weight preserves itself much longer. It can carry and actually absorb a lot more water, helps bring water in and retain water to certain areas, versus the low molecular weight one that'll break down. That's really inexpensive. You can get this stuff online really, really cheap, the low molecular weight ones. They just sell them, but nobody's telling you you're buying low molecular weight HA versus buying high molecular weight. But it's a major component of skin, and it's involved significantly in all forms of tissue repair, what we just talked about, orthopedics, joints, and then we talk about it in the context of skin. It basically deals a lot with balance and hydrodynamics, and actually proliferation and movement of cells from one location to another location. So it helps actually with movements and migration of cells from one place to another place. It's a naturally occurring from umbilical cord stem cells in high molecular weight, based on all the studies and based on the method that we did all the testing on it for us that dates back in 2014. This is once again that diagram where we spent time working with Cleveland Clinic, and these were really, really neat experiments, because when we started looking at how do we produce an abundant of HA, how do we produce an abundant of extracellular matrix, we went down the dynamics of food and how to feed the cells appropriately that were not causing excessive cell turnover to cause the cells to prematurely age, one, and actually cause a grounding or a formation of a matrix so that the cells can be happy and consistently basically produce the right guidance and signaling cues. In this context was HA, registering, even measuring glucose on a daily basis, maintaining blood glucose levels in cell culture in a petri dish on a daily basis to make sure we have the right formulations to get exactly what we have now. So what we told you right now, the product is an abundant in collagen 1, collagen 3, and most abundant in hyaluronic acid, because if you think of the umbilical cord itself, it's a rope. It's a tough rope, and it's predominantly, tissue-based rope that's predominantly hyaluronic acid. But the neat thing about the product is we look at keys to what it produces to be successful in skincare. One of the things that we found, IL-1RA, which is basically a strong anti-inflammatory, FGF2, which is a strong mitogen, helps proliferation, helps basically growth and repair. And then one of the really, really important ones here, and it's crazy, in the appropriate amounts, when you look at this, for those that study peptides, know peptides well, the peptide LL37, which is a natural antimicrobial. And when we start applying this to skin, we started noticing on a consistent basis one clear thing. Those that had blemishes, skin would clear. Those that had acne, skin would clear. Those that had bumps, rosacea, anything unusual within the skin, it would clear. And you basically point to one thing, antimicrobials, which in this context is naturally occurring with LL37. The secretome, what is it put into in the context of our product? It's actually put into a amino acid based formulation that helps stabilize this product. And then, by the way, you guys are welcome to post, which I'm sorry I didn't say earlier, all the questions you want within the chat box there. So go ahead and post away questions. We will get to the questions at the end and go through each and every one of them, because I'm sure there's a bunch of really good questions out there. The question that you don't ask is the question that your partner is wondering about. But please, there is no stupid questions. It doesn't matter. I'm here to answer all of them. But anyway, it's in a amino acid blend formula. All good things: vitamin B12, inositol, biotin, valine, glutamine, glutamic acid, L-arginine, glycine, a bunch of different amino acid blends. The concept is we put it in something that's natural, that helps the building blocks of skin and the building blocks of life, and it actually helps our product maintain itself for a relatively long shelf life, especially the extracellular matrix portions of it. Now, going to what everybody hears out there, those that are in this sector already about exosomes, hear this. They go and send a product out. They go test it on a machine called the nanosite. The nanosite comes back and it registers a quantity of particles. When I first got a report from a colleague of ours that sent us one years ago on our first product line that we actually did, back in, I think it was 2018, 17, around there, we first started looking at testing. We registered something in the range of, it was like 25 billion, it was like 25 billion particles per milliliter without taking much focus on it and then looking at it. And we went back and then said, wait a second, I need to send some different, things out there. And being a scientist, I'm all about controls and controlling experiments the right way. So we know that we're basically doing the experiment and giving you truthful data out there. We sent blinded to a lab, two different labs actually, blindedly some what's called phosphate buffer saline. It's something that we use regularly in the lab. Some Hank's basic salt solution, balanced salt solution that we sent, and even some 0.9% saline. We get registrations back, and then the product also. And of course, the product has a lot more of these nanoparticles. But guess what? These saline solutions had registrations of 800 million per milliliter. One of them had a billion per milliliter. So that tells you right there that this machine is not registering this correctly. So one of the correct methods that we went back and did was using a dye, which is what you see right here, the fluorescence NTA. Because if you look at the product right here, you'll see that, here, right, you'll see 2 times 10 to the 11 in one lot versus the other lot. They're relatively similar. They're pretty stable from production to production. When we produce lots of 20 liters of product that are quadruple sterile filtered. And then you'll see that's why the product is, it's a clear product and it's quadruple sterile filtered. And then we do a lot of testing on these, on each different lot. So, because we have a lot of vials of it. But if you look at this, when they run a standard nanosite, you're talking about, to the 11, you're talking about 200 billion particles versus correctly, this actually says about, we're now talking about to the 10th, we're talking about 50 billion particles or closer to 49 billion particles based on this report, plus or minus error on there. But that is done with a fluorescent dye. And why do we do it with a fluorescent dye? Because this particular fluorescent dye only binds a lipid bilayer. So when you filter everything out, which is done through a 0.22 micron filter 3 times in our case, you now have these nanoparticles that are left over. How do you tell that there's a nanoparticle and not just a tiny something smaller or something unusual that's being registered there? You dye it with a lipid dye, and then it should only register that lipid dye, and hence that's where that number comes from in our product. But not only have we done that, but we've actually looked at how this works on skin in vitro. And what we did here is we've done experiments on taking skin fibroblasts from that of a 25-year-old, taking skin fibroblasts of a 53-year-old, and we grow these up in a petri dish, and we grow up skin cells, and it's a skin model, basically somewhat of a skin model. And then we look at what's inside the cell stored, what's outside of the cell secreted, and we looked at collagen, one, and we looked at elastin. We now actually have some data on hyaluronic acid also. And we only add, in this case right here, we take the cells, we grow them up, we put them in a happy medium, they're happy, they're fed. Then we remove it, then we add the media back in the context of what's called 0%, the product UCM, we call it. Umbilical cord matrix is what we call it right here. And we add it in there, and you will see there's no registered amount of collagen within the cell because we're looking at it in the context of nanograms. There's nothing secreted out or there's nothing stored. Same thing with elastin. But then if we put 5% of the product, so in this case, less is more, or 20% of the product, and then we remove it. So we incubate it for a while. We add it. It's like you're adding it to the skin. And in this case, we incubate it after 8 hours. We put it, on the cells with the food for 8 hours. Then we remove it. We wash it off. Then we put the cells back in their medium, their growth medium, and then we register. What we see is that the product causes your own skin, the own skin cells here, to now produce collagen, to now produce elastin. That's what we want. We don't want something that you just apply to the skin and then you're consistently applying that's helping aid. What I want to do is help something that's going to help your own skin cells, and that's what this does. And not needed in an abundance, because 5% is almost as good as 20%. 20% is better. But if you look at the bars on here, the significant bars on the secreted part, there's not that much of a change, except for in elastin, there's a bigger change on 20% versus 5%. So anyway, ultimately, what we're looking at once again is we want to make sure that the product helps your own skin and your own cells repair itself. Spending time, and everybody's seen there's a company or two out there now producing this in the context of a powdered format, which is basically a lyophilized format, which is easy for transport as one, and two, it's really because the stuff is sent frozen. And two, it's actually really easy because now you can add in the amount of fluid depending on the type of procedure you're going to do, microneedling, radiofrequency, whatever it may be. Topically, you can now add in the amount of solution that you want to bring it down to. We spend years developing this and finding a formula to make sure that this stuff is stable at room temperature. The thing that's helped us a lot is, the product now has trehalose in there. If you look up trehalose, you'll actually see that trehalose actually helps moisture bring in. It's actually really, really good for your skin. It's in a lot of different things. But then we register stability, which is this blot analysis that you'll look to the left with all those bands to make sure, what we have is correct and it's identical to the liquid format, which it is. And then we picked the one with stability testing that has add as basically the proprietary additive formula that we talk about here. And we picked that one and we thought, we actually measured it and we did studies on it to assure that it's the appropriate one that we want to use, which you'll now see right here when you look at induction of hyaluronic acid and induction of elastin. These are the best. This is the best formula right here. That's the one that we now have in a powdered version 2 that gives you, something. So clearing the landscape, it's something that is frequently asked in the context of this entire field. What is going on? I hear from a vendor saying, I have an approved product with the FDA. The product has this in it, the product has that. First and foremost, there are no exosomes approved through the FDA to date. There are some in clinical studies, but once again, if they are in FDA clinical studies, they're not supposed to be sold for anything. Because it's like me thinking of, I have a modified cocaine in a clinical study, and I'm going to sell it on the street at the same time. You cannot do that. It's not an approved drug. It's not approved. Secondly, GMP requires an FDA visit or audit. A lot of people talk about this GMP, and we talk about in the context, we are GMP compliant, meaning we've set up a system that we've gone through all the guidelines, and we think that they're compliant. FDA normally only visits you if there's some form of an issue with a product, an issue with manufacturing, or an issue with something that they've actually seen, where they can come and actually they will come and visit you and they will audit you. A master file is just a document that you file to the FDA. Let's be crystal clear on that. That gives you nothing as it's approved or anything like that. Actually, it's just a document that you file and you register with the FDA. It's now registered. It's nowhere near even having what's called an investigational new drug application, of which our cells have 22 approved to date. The cells do. 22 approved, 4 of them in clinical studies, and 18 of them actually in compassionate use INDs. Cosmetic use is just topical use. You basically look at adding something topically to it. That's what you're using it for. You're looking at enhancing appearance and looking at enhancing appearance, and that's it. Protein is the key to function. Once again, the message does not change into a function unless it's taken into the cell, incorporated into the cell. It needs transfer RNA. Show me there's an abundance of transfer RNA in there that can actually incorporate it, and then show me that it's actually changed into a functional protein. That's what you have to demonstrate. Looking at protein is key in all of this. Claims. The claims are key to everything. You have to be careful with the claims because you can't make drug claims. You're making claims on a skincare that it enhances appearance, it helps with enhancement of appearance, and essentially that's what we're doing. We're trying to make people look better, and when you look better, you feel better. When you walk outside and you look better, you definitely feel better. And everybody knows this when they look at themselves in the mirror. You can tell differences in appearance. whether it's through reduction of fine lines, whatever it may be, um, you know, flushing of skin, plumping of skin, just enhancing appearance is key to what we're looking at and key to what we're doing in here. Use the product. I'm positive and confident you'll enjoy it. It works quickly. What I'm going to do is quickly go through. I'm not a medical doctor. Once again, I'm a scientist. One of my colleagues does a phenomenal job in this, talking about how they actually do this. Microneedling with JuveXO, where they actually use a sprayer. It's like a spray gun that you actually spray the product on. Remember, these are small, tiny particles that if you spray it on, you use less. And then you go through a microneedling procedure. JuveXO can actually get you some of these so you can actually understand how to do this well and actually possibly do some training on them sometime soon. But what you'll see is, a significant amount of improvement. This is doing it on a CO2 fracture laser where somebody had a big old scar because they had a basal cell carcinoma was removed. And what they wanted to do is remove it and make sure that they're not stuck with a scar. Not stuck with a scar, as you can see right here. If they didn't do the procedure, that's a scar that they had before. They did it with the procedure again because the stuff grew back again. And then 2 days post-treatment with JuveXO, you'll actually see clearly that the scarring is actually gone. I'll show you some more images of such following this incredible results. Applying it, this is with microneedling and radio frequency, applying it several times, using once again the sprayer. So you can apply her. It's a big difference in her skin. You can actually tell from somebody that has a ton of sun exposure, as you can see. Stretch marks. This is an individual that they've tried laser, they tried microneedling on its own. Once they apply the product, you'll see a big difference in the stretch marks and everything that was done with it. An eyebrow. This is a weird case of somebody that apparently was hit by a train, had a big old scar on their eyebrow. You can see the scar there. Normally in this type of scar, you will not get hair growth again. They basically actually do microneedling radio frequency, apply the product on there. You can actually see the hair actually growing back on there. And then in hair, we're one of the only ones that have a dedicated hair formula. This is not the same formula that we use for skin. It's a different formula that we developed understanding hair, how hair can grow, how hair can look better, how hair can appear thicker. It's a formula that we actually added 2 recombinant proteins in there, so it helps with the appearance of hair. And you'll see an incredible result right here, before and after. Here's another one, a gentleman that had this done twice, 2 different times. Clearly see quite a bit of differences in it. Works phenomenal. Once again, it's a different formula than the other one. Here's the protocol of it. So you actually have it. You can actually get that from our team. And clearly showing how this stuff actually even helps in the context of, somebody that got a cat bite and the cat bite was infected. Normally this doctor would actually tell her to go to the ER because the swelling of the hand is significantly, you don't want any problem. And then after 2 hours after applying the product, you can see how quick it healed. And you can actually still see the bite mark, after 1 day after, but the healing of the hand, how quick it actually healed. It helps heal skin quite a bit, quite quickly, and it does it fantastically well. Just a basic overview of all of it. Superior exosomes with a strong scientific history. This is made here in the United States, made by us. Contains healing properties. Most importantly, the extracellular matrix is the focus here. It's a key to success in this 3D that we actually live in. HA, collagen, and elastin. Uniform dosing consistently through all the vials, consistently with all the different batches that we've actually produced it. Tested against multiple different pathogens made from umbilical cord lining stem cells that are part of 4 FDA-approved clinical studies and another 18 approved INDs. Thank you very much for your time because I really want to get to the questions. And if you have direct questions, you are welcome to reach directly out to Mr. Brian Pla or myself, Dr. Rafael Gonzalez at JuveXO.com. We are here to help. I'm here to help, and I'm here to help you guys succeed in what you're doing in your practices. Thank you very much. So for the questions.
Thank you so much for joining us today. We appreciate it. If you could take a look at Dr. Rafael Gonzalez on Instagram and take a look at some of his talks. And while you're there, go ahead and follow JuveXO at JuveXO_official. And like Rafael mentioned, give us an email at info@JuveXO.com. And if you have any questions, go ahead and drop them in the chat and we will go through those one by one. The first one, which I think we mentioned, is what about a master file from the FDA? What's the difference?
Well, we talked about that. The master file from the FDA, once again, is just a document that is submitted based on the properties of the product. The FDA does not, look at that and regulate that. It's just a registration of a file. When you want them to look at something, you actually submit what's called the chemical manufacturing and composition in the context of a clinical study. This is basically the full characterization, the full testing, the full everything of the product that's actually done with the product to assure that's done according to guidelines and you've actually tested it. For instance, in some instances, you'll start looking at the potency of it. What does it do? How does it work? What are the pharmacokinetics of the products? Did the product get tested in animals? Did it not? What's the safety profile on it? All of this.
Next question is, what do companies mean when they talk about the number of exosomes in their products?
They talk about normally in the context of the amount of nanoparticles, which is what we talk about. They talk about it has this many nanoparticles. Once again, do not get confused with I have 1 trillion nanoparticles, because if you have a product that's yellow in color, any other color, this laser registers through there is going to get a backdrop of dye of the color, and you're going to get incorrect counts. And once again, just like I told you, I can run saline that's crystal clear through this machine, and it'll register 800 million particles. I wouldn't focus on that as much, even though the product we have has a significant amount of exosomes. I'd focus on the protein concentration of what the product contains.
Next question, what's the added proteins in the hair product, and what's the difference between the hair product and the skin product?
I would love to tell you that proprietary blend, but I cannot. I'd get in a lot of trouble if I did, and my other scientists that work with me would really hate me. But anyway, it's a proprietary blend of 2 recombinant growth factors that we've actually added in that are designed to help, hair look really well, grow. Designed with science. There's a lot of science behind them. A lot of research we actually done with them.
Next question, out of curiosity, the first medium on the chart seemed to result in more HA and CS cell 1. Why wasn't that one used?
That's a great question, and we're going to go back to that chart so I can, let me pull that chart up for you. Let's pull that chart up. Now, just so you understand, that is what's inside the cell. Okay? UC number 1 is what's inside the cell. What we're trying to capture is not what's inside the cell. What we're trying to capture is what's outside the cell. I'd have to disrupt the cell, capture everything that's inside the cell. And then what we look at, if you look at what's UC1, UC cell 1 versus UC media 1, the majority of it was sequestered and maintained inside the cell, and it wasn't released by the cell. What we're looking at more of what's outside of the cell, what's something that's better that's outside of the cell. So remember, the cell is the machinery that produces it, and then it releases it out into the environment. The environment is, in this case, the media.
Next question, what is the difference between lyophilized and the regular skincare formula?
Great question. So one of them is actually in a liquid format, pre-made liquid format. All of it's done in a liquid format to start off with. Lyophilized is basically the way the product we made it is. A lyophilized product that we have is basically in a 1 ml format. Just 1 ml of our product, lyophilized, put into powder. Makes it easier for shipping, makes it easier for storage. And then it actually puts it into context of, for instance, if you need to take that 1 ml and dilute it down, if you wanted to dilute it down to have the spray a little bit more spraying capacity, you can do so. You'll be surprised. I showed you images of less is more in our studies. You can dilute the stuff down in simple saline solution, 0.9% saline solution, and it works great. And you can use more, you can spray with more frequency as long as you get some product through the skin.
Perfect, thank you. Are there any other questions that you'd like to ask?
Thanks everybody for your time. I appreciate you once again. Please do not hesitate to ask other questions offline, whatever you like. We are here to help.
And please stay tuned. This is the first in our webinar series, so we'll be sending you more information soon. Oh, and we actually had one more question come in. Is microneedling considered topical?
I mean, it's essentially microneedling because all you're doing is you are not injecting this in. You are spraying this on. Remember that. You're just spraying this on. That's all you're basically doing. You're basically taking it, spraying it on, and then after you spray it on, you will microneedle it. After you microneedle the product, then you can spray it back on. You're not needling it in directly. You're not doing anything to inject it in to penetrate the skin. The device is the device.
Any other questions before we go? So it is not cleared for injection at all.
Correct. Actually, there's really nothing out there except for some HA, some filler products that are cleared for injection. Thanks, everybody. Have a wonderful evening.